Okay, we have come to the last chapter of Biology Form 5, and that is chapter 13, Genetic Technology. Okay, in this chapter, there are two subtopics. The first one, Genetic Engineering and Biotechnology. Okay, so first, let's look at the definition of genetic engineering. Okay, genetic engineering is gene manipulation technique to modify an organism's genetic material to produce new combination of genes. An organism such as animal, plant or microorganisms that is produced by recombinant DNA technology is known as genetically modified organisms, the GMO. So we have GM microorganisms, GM animals, and also GM plants. Genetic engineering involves the transfer of DNA segment from one organism to another by DNA recombinant technology. By using the techniques and procedures in the recombinant technology, biologists can recombine the DNA or genome, referring to a complete set of DNA of an organism. Okay, here is the definition of genome, a complete set of DNA for an organism that includes all the genes of that organism. The genome contains all information needed to build and carry out life processes of an organism. Now let's look at the basic technique of genetic engineering or gene manipulation. Okay, there are basically four key steps, which the first one involves cutting out DNA of the desired gene. So here the cell carrying the desired gene is broken and its DNA is isolated from other components of the cell. And a restriction enzyme is needed. It works like a pair of scissors to cut out the desired gene. The second step is inserting the gene into a vector. So the gene of interest is put into a vector which acts as a carrier to carry the gene into a host cell. And the gene is inserted into the vector with the help of DNA ligase. The third step is introducing the vector into the host cell, where the host cells allows the vector to replicate, and the host cells can be bacterium, a yeast, or even a mammalian cell. Okay, the last key concept of gene manipulation is multiplying the copies of the gene by the host cell. As the host cells divides, identical copies of the gene of interest are also produced in the new cells, which the new cells are then able to synthesize new protein.